Okay, so welcome everyone for this last but not least on Helix Innovation Workshop that we highlight some of the great work that ARU and the Arise Innovation Hub has done in terms of COVID-19 research. So I'm delighted to be joined today by one of their researchers who will present on his own work, but also show the work of one of his colleagues, and he will explain to you how to get in touch with him. As usual, it's a workshop, and so we will welcome any comment or questions that you will have just to put in the chat box, and I will pick up on them, or speakers will directly, and so just to make it a bit more interactive, just letting a few more people in. So without further ado, I just want to leave the floor to Beverly, who will give you a quick introduction of ARU and Arise and pick it up from there. Thank you, Beverly.
Thank you very much, and lovely to be with you this afternoon. As kindly, the slide that's gone up, we're really proud to sponsor this session, this workshop this afternoon. I'm here as the Innovation Hub Director of the Arise incubation sites, which are split across Chelmsford and Harlow and are part of Anglia Ruskin University. So the Chelmsford Hub is located on the campus where Selim and Stephen are based, who will be speaking this afternoon. And our new Harlow campus opened in August 2020. I joined the team last Monday, and I'm just getting my feet under the table. Our innovation hubs are looking to really maximise the potential that we have in the Essex region around health tech, med tech, and well-being, and align with the research expertise that we have within our own university at Anglia Ruskin University and regionally. And I think under that, we want to really look at how we create this really buoyant knowledge economy. And so this afternoon, we've got two presentations showing some of the research excellence that we have. The first will be Selim, the second will be Selim, and the first will be Professor Stephen Bustin, who's recorded a session for us today. And if you have any questions on the presentations, or you'd like to get in touch about what the innovation zones look like and what engaging with our medical research teams looks like, please do get in touch with myself or Selim, and we'll be more than happy to talk to you. So without further ado, I'm going to give a nod to Selim to get our video up. The first of which is looking at some of the novel and cutting edge diagnostic capabilities of Professor Stephen Bustin's lab and how they've responded to the pandemic year and rapid PCR for COVID. Over to you, Selim.
Thank you, Beverly. So Stephen has sent his apologies. He couldn't be here today, so he sent his pre-recorded presentation. So if you have any questions to Stephen, I'm going to put his email address in the chat box. Please feel free to email him directly. He will be more than happy to.
Hello, everyone.
It does.
Simple, accurate, and fast
Enjoy.
testing of large numbers of individuals for the presence of pathogens is one of the most important weapons in our public health arsenal. The current pandemic has also revealed it to be a fragile, often misapplied, as well as error-prone tool. COVID-19 testing generally involves samples obtained using invasive methods, most often nasopharyngeal swabs. This is followed by a complex logistical operation before viral RNA can be analyzed and a diagnosis is established. The analysis itself can be done using a variety of techniques, some more useful than others. For now, CRISPR is still too experimental to have any practical impact. Sequencing is certainly not fast nor inexpensive, and neither is digital PCR. Both, of course, have roles to play, just not as viable methods for cheap and rapid mass testing. LAMP is complex, and at around 20 minutes is not what I would call fast. This leaves PCR, which despite being around for 35 years or so, is nowhere near having reached its true speed potential. So the aim of today's presentation is to communicate our approach to simplifying and speeding up pathogen testing by using the example of SARS-CoV-2 and the diagnosis of COVID-19. A useful testing protocol requires optimization of the diagnostic workflow. Hence our focus on pre-analysis as well as various areas of the RT-qPCR method itself. And today I shall discuss briefly two of these. The first is aimed at streamlining and simplifying the sample collection and extraction process. The second involves developing extreme PCR protocols and devices on which to test and eventually run them in a diagnostic setting. The methods currently used to obtain samples for testing, at least in the UK, appear designed to make the process as cumbersome, unreliable, inefficient, and easily contaminated as possible. Clearly, our current pre-analysis workflow is not fit for the purpose of rapidly communicating a reliable test result to a potentially infected individual so as to minimize the chance of further transmission. Instead, results take hours, if not days. Large laboratories can easily spread contamination, and the efficiency of sample collection and processing is highly variable. One obvious way forward is the development of a point-of-care solution that combines the collection process with an enrichment step. For most current SARS-CoV-2 variants, the obvious sample source is saliva, and although this has been obvious since at least February 2020, it is extraordinary that most protocols still use swabs, often taken by inexperienced and hence unreliable self-swabbing. Eventually, we envisage a device that will combine sampling, amplification, and diagnosis. Just as importantly, I see the integration of RNA extraction and of DNA generated using random primers as an important approach to maximizing sensitivity. This will allow separate optimization of the RT and PCR steps, as well as easy subsequent analysis of samples for other target sequences, for example variants. The RT reaction is surprisingly resilient and can tolerate lower levels of saliva without massive loss of sensitivity. As you can see here, whilst 5 to 15% saliva in an RT-qPCR reaction leads to some decreased sensitivity, it also suggests that a minimal cleaning up process could eliminate this inhibition. This is relevant when aiming to minimize the number of washes that need to be carried out prior to the RT. I must stress, of course, that RTs do differ in their tolerance. The PCR reaction can be even more tolerant, obviously dependent on the choice of polymerase. With this one, there is a twofold decrease in sensitivity in the presence of 10% saliva. Obviously, judicious choice of both Rosen scriptase and Taq polymerase is important. I should also add that we have found that the consistency of saliva collected from different individuals differs considerably. Our method makes use of streptavidin-coated dynabeads, which have biotinylated reverse-sense oligonucleotides targeting viral and human RNAs. Heated saliva is added to the dynabeads, and the solution is briefly vortexed. It is then incubated at 30 degrees centigrade for 2 to 3 minutes, and then placed near a magnet. The supernatant is pipetted off, and with or without a wash, a 1x RT mix including random primers is added. Samples are incubated at 55 degrees for a minute, and then added to a PCR reaction, with specific primers and probes obviously. At the moment, this process takes 5 minutes when done manually, but the potential for more rapid protocol is obvious. Here's an example of the kind of initial results we have been getting. As you can see from the CQs, results depend on the RT, the PCR master mix, as well as the target. But we have been able to achieve delta CQs of around 2, i.e. a 4-fold loss of sensitivity compared to pure, unextracted RNA. Given that we can enrich to a far greater degree, we feel this is an encouraging result and a useful starting point. We have not yet optimized the lengths of the biotinylated probes and determined the optimal binding density for the oligonucleotides. More recently, we have been achieving even better results by including Takara's sample prep solution with the saliva and using it as per their protocol. As you can see, the delta CQ between extracted RNA and pure RNA can be as little as 1.2 in this RT-qPCR experiment. These data show results obtained using two different RT-qPCR master mixes. Master mix A in blue is less inhibited by saliva than master mix B in pink. Both are as efficient at reversal and striving at 1 minute as they are at 3 or 5 minutes. For master mix A, a CQ of 25.32 at 5 minutes compares to CQs of 25.53 and 24.90 at 3 and 1 minute respectively. Also note that this is the case without having to increase the amount of RT in the reaction. So in summary, we are able to extract, enrich and generate cDNA from saliva in around 5 minutes. This can then be used in a separate qPCR reaction. The RNA can of course also be processed using a one-step protocol. We have also had success with using biotinylated oligos to capture viral RNA and then use streptavidin-coated plates to isolate and reverse transcribe that RNA. The challenge now is to optimize the various ingredients and incorporate them into a device, whether a point-of-care or extraction robot, that obviates the need for complex pre-analysis steps. Our second focus is on speeding up the PCR reaction. Carl Whitwer demonstrated more than 6 years ago that a PCR run can be completed and analyzed in real time in around 17 seconds. We have published several papers that demonstrate the use of 1 second denaturation polymerization times. Nevertheless, the speed potential of PCR has never been properly exploited, and although there is an instrument that can carry out PCR reactions in less than 10 minutes, it requires proprietary plastics and, from what I can see online, does not use an extreme PCR protocol. We are now investigating the many variables associated with developing extreme PCR protocols. We are doing this using a bespoke robot cycler which grants have made for us. The instrument consists of 2 water baths, one set between 60 and 70, the other between 95 and 97 degrees, and a robotic arm that dunks reaction vessels between them. Denaturation and polymerization times are infinitely flexible, as is the speed at which the arm travels between the 2 baths. At its fastest, which means dipping in each tank without holding, the instrument can complete 30 cycles in just over a minute. We also have developed a range of SARS-CoV-2 assays, some of which have been published, although they were designed with fast rather than extreme PCR in mind. I define an extreme PCR as one that can complete 30 cycles in less than 3 minutes, whereas fast PCR takes around 15 minutes. This video demonstrates part of a 1 second denaturation, 1 second polymerization run, which takes just over 2 minutes. You can see that there is lots of potential for speeding up the process by using bespoke water baths rather than 5-litre ones, and by placing them closer together and making the robot travel faster. This is all a matter of cost, obviously. We are testing a wide range of plates, tubes and strips as they all perform differently under the extreme conditions we apply. Obviously, we need to find a combination of small reaction volume in the smallest vessel possible to minimize condensation. At the same time, the plastic needs to be thin to maximize heat exchange. The robot cannot detect fluorescence yet. Hence, we are monitoring amplification either by comparing fluorescence units using a qPCR instrument before and after amplification, or we use a workaround that involves transferring an aliquot of the amplified target into a standard qPCR master mix and running it on a qPCR instrument. We are looking for a shift in CQ compared with an unamplified control as per amplification plot at the bottom here. This gives us a measure of how efficient the extreme PCR amplification is, and in theory, each cycle of extreme PCR should reduce the CQ in real time by 1. Our immediate aim is not for real-time detection, as we feel that initially a yes-no answer is sufficient and will be a lot cheaper to implement. But again, Carl Whitwer has demonstrated that real-time fluorescence detection of extreme PCR reactions is perfectly feasible. Carl provided a number of guidelines to optimize extreme PCR, and high primer concentrations is one important parameter. If you look at the left, you see results from a Saliva green assay. It was carried out using a published SARS-CoV-2 assay targeting the polymerase gene. Unfortunately, increasing primer concentration, for example 5-fold, can result in significant increases in the amount of non-specific amplification in the no-template control. However, if you look at the right, then the same target is being detected by a probe, and this becomes a non-issue. Here we are evaluating the effect of using very high primer concentrations. The target is variant B117, which does not have a mutation at the EEC codon. The wild-type sequence is detected by the FAM probe in blue, with the hex-labeled mutant probe in green not detecting the PCR amplicon. After 20 cycles, the standard primer concentration of around 300 nanomolar, A here, results in a delta CQ compared with the unamplified control of around 3. At the optimal primer concentration, 10 preamplified cycles increase the delta CQ by 10 after 10 cycles and by 20 after 20 cycles. Reducing the denaturation time to 1 second initially resulted in less efficient amplification, with the actual impact depending on the assay. As you can see, when amplifying B117 cDNA, the CQ for the wild-type assay, again in blue, is now 10 cycles, when at 20 seconds it was 20. When targeting B1.351, the mutant assay has a delta CQ of around 8, again the expectation being 20. The only difference between the two assays, remember, is a single nucleotide in the middle of the probe. This suggests that probe design is another parameter to consider when optimizing extreme PCR conditions. If run for a full 30 cycles, this run would take 2 minutes and 15 seconds. Reducing the polymerization time also affects the results, but then these results were achieved without increasing the polymerase concentration, another parameter Carl Witwer has highlighted as important. Also bear in mind that some polymerases are faster than others, and certainly the aim is to develop a protocol that uses 0-second dwell time at 60 degrees centigrade. I suspect that this will be easier to achieve than reducing the denaturation time significantly below 1 second in this setup. The runtime here for 30 cycles was just over 2 minutes. The reaction vessel has a huge impact on the success of the extreme PCR. Here's an example where the use of a standard 96-well qPCR plate from a major manufacturer does not result in efficient amplification. The delta CQ after cycle 20 compared with the non-amplified control is only around 5. Also, with most standard qPCR plates and tubes, it seems that larger volumes, around about 15 microliters or so, work better than smaller volumes. However, we have now identified a suitable reaction vessel where this does not seem to be the case, and where smaller volumes amplify more efficiently, as you can see for the wild-type assay here in blue. Nevertheless, this requires further investigation, as the results for the mutant assay are not quite as clear-cut. Clearly, it is important to identify not just optimal reaction volumes, but also optimal reaction vessel volumes for extreme PCR to work efficiently. These are results from an experiment with 18 replicate amplifications, which took 2 minutes and 27 seconds. Reactions were diluted 1 in 1,000, and aliquots from each reaction were used as templates in standard qPCR reactions. The mutant B1351 K484 allele is detected using a hex probe. In pink, you can see replicates of neat, unamplified cDNA for comparison. As you can see, there's consistent detection and good uniformity. Finally, a comparison of two master mixes shows differences in the efficiency of amplification. Again, aliquots of diluted reactions were re-amplified, and the delta CQ between master mix A and B is the equivalent of about a 250-fold difference. So to summarize, obviously we are only at the beginning of our investigations, but we have established conditions that result in 30-cycle runs with efficient amplification of a dual-plex assay in around 2 minutes. This was achieved using assays not yet optimized for extreme PCR. Of course, we have only started to learn what the critical assay design parameters are that allow successful extreme PCR runs. But clearly assay design will be important, as is primer concentration, choice in polymerase concentration, and reaction vessels. We have tested a range of magnesium concentrations though, and these made no obvious difference. In conclusion, there can be no doubt that PCR-based methods offer the best prospect amongst nucleic acid testing methods for immediate, accurate, flexible, and cost-effective implementation of mass testing. The key will be to integrate sample collection, preparation, and analysis to minimize the risk of contamination, variable sample quality, and so the risk of false positive or indeed false negative results. This leaves only the question of how to interpret a positive test result within a clinical context. But that is a question for another day.
Right. So I'm going to write Steven's email address in the chat box. So if anybody has any questions, please feel free to email him directly. Just mention that you listened to his talk at the One Helix meeting, so he can make the connection. Great. So let me introduce myself. So I'm Selim Cellek. I'm the lead of the drug discovery group in Medical Technology Research Center at Anglia Ruskin University. And I'm going to talk to you about our work on development of antifibrotic drugs. So if you have any questions, please feel free to put them in the chat box, or you can ask after the at the end of my talk if you got time left. Right. So I'm going to talk about the irreversibility of fibrosis, and particularly COVID-related fibrosis. So first of all, fibrosis, it is one of the biggest killers in the world. It is estimated that fibrotic diseases account for 42% of mortality globally, and the main killers are liver fibrosis, which also known as cirrhosis, and most common cause is alcoholism. Heart fibrosis, cardiac fibrosis usually happens after myocardial ischemia. Kidney fibrosis is also known as chronic kidney disease, and most common cause is diabetes. It's one of the most common diabetic complications. And lung fibrosis can be in different forms, but well known is idiopathic pulmonary fibrosis. And you can see some numbers here and the mortality. The thing is, fibrosis can affect any organ or tissue in our body. And here are examples of some common fibrotic disease. Peyronie's disease, which affects the penis, which causes curvature and erectile dysfunction. Dupuytren contracture causes disfiguration and dysfunction of the fingers, more common in men than women. Scarring that happens after burn injury, either chemical or heat burns, or surgical or trauma injury to the skin, and that is fibrotic condition. And intra-abdominal adhesions that happens after radiotherapy or surgical intervention, and which may require further surgery to release the adhesions. Again, this is fibrosis. So we all have these fibrotic diseases to deal with in the clinic, and situation is actually getting worse because COVID-19 are now known to cause pulmonary fibrosis, lung fibrosis, heart fibrosis, and kidney fibrosis. And very recently, some data started to emerge that COVID-19 may also affect the liver, cause liver injury, and that may lead to liver fibrosis. But it's very, very early days for anything to say about liver. So I just wanted to give you some examples of the impact of COVID and fibrosis. So it is estimated that approximately 40% of patients with severe COVID illness develop ARDS, which is acute respiratory distress syndrome, and 20% of these cases are severe. And of these ARDS due to COVID who are discharged from the hospital, meaning the ones that can survive, 30% develop lung fibrosis. And 10 to 15% of patients who were hospitalized with COVID are expected to develop lung fibrosis in the next couple of years. So if we know that 500,000 patients in the UK were hospitalized because of COVID-19, and if we take 10% of that, that means approximately 50,000 patients with lung fibrosis will need to be taken care of by NHS in the coming years. And this is not unique to COVID-19. Do not think that this is lung fibrosis can be caused only by this virus, but actually other respiratory viruses like SARS coronavirus, influenza H1N1, and MERS, they all are known to cause lung fibrosis. And the problem is, and that is the message I want to give, is fibrosis is irreversible. It's progressive and it's chronic. And that's what I want to dwell into and share some of our research with you. So probably you are wondering what fibrosis is. So I want to share you an animation. It's a 1-minute video. And this animation was made by one of our ARU's art design students, undergraduate students. And I think it explains very well and it sets the scene. So if you bear with me, 1 minute of animation.
Worldwide, fibrotic diseases are responsible for 42% of all deaths. But what is fibrosis? Fibrosis happens when our body's natural ability to repair itself goes wrong. When an injury occurs, the body sends fibroblast cells to help with repairs. But when they are dispatched to a wound, they transform into myofibroblasts, which are supercharged fibroblasts. Sometimes, instead of closing the wound and dying away, they start overhealing, resulting in excessive scarring. This out-of-control wound healing is fibrosis. When this scarring happens in a vital organ, it can cause a lifetime of health problems and can be fatal. A major organ commonly affected by fibrosis is the lungs. The lungs are made up of millions of tiny air sacs. If the thin walls of the air sacs become damaged, scar tissue can build up over time, making it difficult for oxygen to get into the bloodstream. The scar tissue makes the lungs stiff and they are less able to expand, making it difficult to breathe, and without a lung transplant it can be fatal. Cardiac fibrosis can be triggered when the heart muscle is damaged from heart disease or a heart attack. The heart muscle becomes stiff and loses its power to pump blood around the body, resulting in heart failure. Fibrotic conditions can also lead to kidney failure and liver failure. There is very limited safe treatment to manage fibrotic conditions, and often a transplant is the only hope. However, research is being done by the Medical Technology Research Centre to find new and effective drug treatments. Worldwide.
Oh, sorry.
Worldwide, fibrotic.
Okay, so it is scarring. It can affect any tissue, and it is the aberrant wound healing. And the myofibroblasts is the key cell. And this irreversible and progressive proliferation of this myofibroblast cells causes accumulation of extracellular matrix protein that causes tissue dysfunction and organ failure. So that is the main crux of the pathophysiology of fibrosis. So what have we been doing in the last 5 decades to find drugs for fibrosis? First of all, just to summarize, there are only 2 drugs, and that they are for lung fibrosis, and they got very limited efficacy and safety. And there is no other drug for any other fibrotic conditions. So in the last 5 decades, pharmaceutical companies have been selecting individual targets like TGF-beta, IL-13, PDGF, and developing inhibitors or antagonists or ligands to interact with these targets, hoping that when you inhibit, for example, PDGF, it's going to prevent or reverse fibrosis. And all of these drugs, and the list actually is longer. Only some of the drugs that have been developed are shown here. They all failed in phase 2 clinical trials. So after 50 years of research and billions of dollars of investment, we got no drugs whatsoever. Why? Before I answer that question, just summarize the current pharma landscape. 2 FDA-approved drugs only for idiopathic pulmonary fibrosis, as I mentioned. Their efficacy is very limited. Currently, last year, there were only 15 phase 1 trials, 57 phase 2, 13 phase 3. They're all single target compounds, meaning compounds that are acting on a single target. And they all fail or about to fail in phase 2 due to lack of efficacy. So what we are suggesting is maybe this approach for in the last 50 years is wrong. Maybe we should try different approach, which is called phenotypic screening. So what is phenotypic screening? So this is single target approach. You select a target like PDGF receptor, and then you screen millions of compounds against this target. You find a hit, and you develop that for clinical trials, and it fails. But there is another approach, and this is actually older than the single target approach. You choose a phenotype instead of a target. Could be a change of a cell, a typical cell, or critical cell, or a function, and then you screen compounds against this phenotype, and then you take your lead compound. And actually, this approach has been shown to be more effective than single target approach. Here is the first-in-class drugs. 37% were discovered by phenotypic screening, and 23% were discovered by single target approach. So phenotypic screening has been shown to be more successful than single target. And so we decided to take this approach to find new antifibrotic drugs, and the phenotype we are targeting is the myofibroblasts, because that is the culprit. That is the cell that causes fibrosis. So how we are doing this? So we first decided to use human primary fibroblasts from patients with fibrosis. So after patient recruitment and consent, we take this relevant tissue from the patient. We isolate primary fibroblasts, and in a 96 well plate, we transform them into myofibroblasts, and we developed a medium high throughput phenotypic assay with a good reproducibility to be able to test compounds. So what we do is the human fibroblasts we isolated from the the patient. We give them the cytokine to transform them to myofibroblasts, and that is the cell causing fibrosis. And at the time of the transformation, we add compounds, drugs, whatever you want to test, to see if those drugs, compounds will inhibit the transformation. And that is the basis of our phenotypic assay. Is a typical 96 well place. So these are the cells, these are the wells that contain cells with the transformed myofibroblasts. They are green, here they are. And these are the control cells that did not transform. And the reproducibility is quite good. So what have we discovered so far? And this is just the validation of our assay, showing that an old Smith-Klein-Beecham compound, which is a TGF-beta-1 receptor antagonist, is able to inhibit the transformation as a positive control to validate our assay. So first disease we decided to tackle is Peyronie's disease, the one I mentioned, the fibrosis of the penis, and we have screened just under 2,000 FDA-approved drugs. We got 41 hits, and then 11 hits from 6 classes shown to have desirable safety profile. And then from those hits, we identified 2 classes, phosphodiesterase type 5 inhibitors, PD5 inhibitors. The most commonly known one is Viagra, and selective estrogen receptor modulators, and the most commonly known one is tamoxifen, which is used for breast cancer or osteoporosis. So, and interestingly, when you combine these 2 classes, they synergize in vitro and in vivo. And we have published that, and we just filed a patent on that. So we have 2 classes of well-known drugs which have been licensed for other indications: PD5 inhibitors for erectile dysfunction, and SERMs for osteoporosis and or breast cancer. And we are trying to repurpose them for treatment or prevention of Peyronie's disease. And the second disease, fibrotic disease, we targeted is the scars, the skin scars. So we again isolated fibroblasts from burn patients, skin of the burn patients, and we did this exactly the same screening, again same FDA-approved compounds, and we got 19 hits. But then when we selected them or triage them according to their safety and topical application, we decided that 2 of these drugs are worth taking forward. We just filed a patent on that, and we are hoping to reposition this topical agent for prevention of skin scarring. So this is our group's pipeline: Peyronie's disease, phase 2B in progress, skin burn. We just filed the patent. Lung tissue, we are desperate to get some lung tissue, and then so we can do the same. But as you can imagine, it is very difficult to get COVID lung tissue due to COVID-19. But we got intestine and heart tissue, and those projects are in progress. So let's go back to the main question: is fibrosis reversible? So with the exception of mild to moderate liver cirrhosis, fibrosis is irreversible, progressive and chronic. And the reason for liver being the exception is because in mild to moderate liver cirrhosis, liver has the ability to regenerate, but all of our other organs do not have that ability. And actually, we have shown that the myofibroblast transformation becomes irreversible in vitro in 24 to 36 hours. And if you are interested, the paper is published there. So we have a very, very narrow window of opportunity to prevent the progression of fibrosis. Once it is established, it becomes irreversible. Can it be prevented? Yes, if the disease is diagnosed early, the progression of the pathology can be halted or slowed down. And to prove that point, I want to show you some clinical results which we got very recently and we have not published yet. So this is a phase 2A study in early Peyronie's disease. Our colleagues at UCLH have done that. So when we catch patients with early Peyronie's disease, so the fibrosis has not established itself, we gave them either no treatment or vitamin E, because that's all we can give, or we gave them off-label tamoxifen and a PD-5 inhibitor. And the controls that receive no treatment of vitamin E are in blue, and the ones which receive tamoxifen and a PD-5 inhibitor like Viagra or tadalafil, etc., they are in orange. And as you can see, control group majority, they within 3 months they got worse or they had no change. However, the ones with a treatment, they improved or they had no change. And the ones that improved, as you can see, the difference is significant. And our statistician tells us the treatment, there's a significant association between treatment groups and outcomes. So this is unpublished phase 2A study. It's a retrospective observational study. It's not randomized clinical trial. With all the caveats, I think we strongly believe that there is a good clinical signal. That's why we are trying to do phase 2B randomized clinical trial. So in conclusion, we have developed a high throughput phenotypic screening assay, which can be used for any fibrotic diseases. And so far we have screened FDA-approved drug library in Peyronie's disease and skin scarring. And further screening campaigns are planned for heart, intestine, lung fibroblasts. And the hits we identify are progressed to in vivo animal models and clinical trials. So with this clinical data, we have some now assurance or confidence that phenotypic screening approach might be the way forward to identify future antifibrotic drugs. I think this approach will have a better chance of success than single target approach. But the new antifibrotic drugs are very unlikely to reverse fibrosis, but they do have the potential to halt progression if they are given early. So COVID-19 is going to have a huge impact on healthcare systems across the world in years to come. So we need to really accelerate our efforts to find novel drugs or repurpose old drugs for fibrotic diseases so they can be administered to patients who have survived COVID-19. I just want to acknowledge my colleagues, my former PhD students and current PhD students and research fellows at Anglia Ruskin University, our clinical colleagues at Broomfield Hospital and University College London, the funders, and of course all those patients who have donated their tissue. So I'm more than happy to answer some questions. I think we got around 8 minutes left. Thank you very much.
Thank you, Selim. It was a really impressive presentation. I can't see direct question in the chat box. So I'll just kick off with one because obviously it's, as you said, very important condition, poorly addressed, also maybe difficult to understand the reason why. But now it's been more under the spotlight because of the COVID consequences. What are you calling now? Is it more public interest? So I don't know, more government funding in that area, or is it more industry interest? And so you're looking for partnership with industry?
So I can tell you where we are stuck. So when we screen FDA approved drugs, all of our hits are now generic. They are off patent. And generic drug companies have no interest whatsoever repurposing generic drugs because they are saying they don't have any incentive to pay for all the cost of repurposing. And these are legal, regulatory, clinical trial costs, etc., etc., etc. And there's some considerable amount of cost attached to that. I can understand them. So they are saying they don't have, there is no incentive for them to do this, and they are not a charity. So there's a gap there, and NHS England has actually recognized that gap, and they are working with the generic drug companies and life sciences department try to close that gap. So that's a big gap. If we need to bridge that gap, either through governments or charities. So generic drug companies need to be incentivized to take these drugs forward. Drugs are there, they are safe, they are ready to go. It's just a matter of funding putting in place.
Yeah, I think it's funding or reassurance that the market is there. So you're saying the patients are.
Patients is there, market is there. I mean,
I think by market I mean is having reassurance, for example, with the NHS or the country, health systems to say, yes, we see that there are definitely a clinical unmet need here, and we like to support, with regulatory support or other, and make this drug going to market and pay for them. I think that's the big gap. That's what I'm saying by making the market. Okay, so yeah, I hope the call is out now. Sorry, Barbara, I can see you have your hands up.
No, I just wanted to thank both Stephen and Selim for fascinating presentations. Selim, I wanted to ask you whether age of the patient makes a difference because of the natural repair mechanisms within the body. I know you're talking, this has to be done obviously early because if fibrosis is advanced, it's very difficult to treat. But I just wonder whether age of patient comes into it because the speed of fibrosis is faster, whether the reaction to the drugs is better or worse or whatever.
So only data we have so far is in Peyronie's disease, where we have observed that the fibroblasts from elderly gentlemen are slower to grow and slower to transform. I mean, that is not unexpected because there's cellular senescence. But from the clinical data, we haven't seen any age difference. But again, the numbers are very small. But it can be a contributing factor, definitely.
Thank you, Barbara and Selim. So if there are any further questions, now is the time. Or if not, so you have Stephen's email address to ask any question relating to this COVID testing technology and development. And Selim, if you're happy to also be contacted for any further information. I think there's just, yes.
I'll put my email address.
Absolutely. And obviously, this has been recorded and so will be available on demand. So if you want to share with any colleagues, I think to make it widely available would also be a good idea because, as you said, Selim, it's a call for collaboration and support to address this very important unmet medical needs. So I would be very, very happy to support that, of course. And I think if I don't see anything popping in the chat box, I will thank you for your time. Well, it was really good to have both perspective of the therapeutics angle and the diagnostic space, which really shows the breadth of what Anglia Ruskin University is doing in terms of research and in the answer to this COVID-19 crisis, of course. So a big thank you to you. Thank you, Selim, for having covered for Stephen as well. It was really good. And thank you very much, Beverly, for joining and saying a few words about it. Is there anything you would like to add on how to, I know you covered, but how to interact with you, if you're also happy to be contacted, if people want any more information?
Very happy to be contacted. My email address, we can post after the meeting, but it's beverly.vaughan@aru.ac.uk. And if there are any points that Selim or Stephen has raised in terms of, you're interested in working with that diagnostic capacity, or whether it be working with Selim on looking at repurposing novel compounds, accessing those phenotypic screens that he mentioned, we'd love to hear from you. And if you are looking for where to find a home as a small business in this field, again, have a chat with us about the Arise Hubs.
Thank you very much. So I am cutting it an end, and thank you very much for attending. It's also the official end of Helix, and so we'd encourage everyone to have a look at all the on-demand content, and also the app it remains open until the 3rd of August. So if there is anyone you would like to contact through this app, you still have a few weeks to do that. So thank you again.
Thank you very much.